feeling like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong, in my happy place. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong, in my happy place. And I'm right where I belong, in my happy place. And I'm right where I belong, in my happy place.
Hello, this is Brian Kennedy. Welcome to the National University of Singapore Yonglou Lin School of Medicine's Healthy Longevity webinar. Thanks for joining us. I have to say, if my voice sounds a bit raspy tonight, there's three reasons for that. One is that I had a virus recently. A second one is that I'm coming to you at 3 in the morning from Vancouver, Canada, where it's nice and crispy cold here. And thirdly, I just got through recording about 4 hours of video with Matt Caberline for his webinar at OptiSpan in Seattle. And whenever Matt and I get on the same video, we yell and scream at each other so much we lose our voice. So I will announce that where that webinar is available to the public next week on the show. And you can go on and see us answer about 100 different questions about anything to do with longevity. Tonight we have Myriam Grunewald with us, and we're really excited to hear her talk in a few minutes. Please use the Q&A function to ask as many questions as you like, and we'll try to get to those as we can. Before we do that, though, we have Mohammed Shafiq, who's a research assistant at the NUS Center for Healthy Longevity, and he'll be presenting on the center's clinical studies, functional exercise, a leg extension test. Thank you for joining us again.
Thank you very much for having me. As mentioned, yes, I'm Shafiq. I'm the exercise physiologist, and today I'll be presenting on the functional exercise 8 repetition max leg extension test. So for today, we'll be mentioning about the why are we doing this, what is the test about, how is it measured, the exercise guidelines, the reliability across both genders, and lastly, the summary. Now, we all know that muscular strength is associated with health outcomes. In 2001, Kell and colleagues mentioned that low muscle strength was independently associated with elevated risk of all-cause mortality. Now, if we were to exercise regularly for strength training, then the risk of coronary heart disease will be reduced significantly. There will be an increase in bone mineral density, meaning to say the risk of having osteopenia or osteoporosis will be reduced for the aging population. There will be an increase in flexibility, increase in glucose tolerance, and as well as for the activities of daily living, meaning the quality of life. Now, what is this test about? Simply to put it, the individual will be seated on this stacked machine, which you can find it either in the gym or the physiology lab, and will be performing a leg or knee extension outwards. The primary muscle group would be the quadriceps femoris or the rectus femoris, in particular vastus medialis and vastus lateralis. Now, there is a exercise guideline to adhere to prevent from any injuries. So firstly will be the proper technique. Now, to perform the proper technique, make sure the individual will be rested nice comfortably, the ankles behind the foam padding, hands by the side, head in neutral position. As we perform the exercise, we would like to do a proper breathing control, meaning to say, as you exhale, you would like to exert the force outwards. So inhale first and then exhale, exert the force and then inhale back, and it becomes a cycle. Doing the full range of motion will be great. We do not want to either hyperextend or perform a half rep, then it wouldn't count. Lastly, selecting the appropriate weight will be great. We want to prevent from any injury if you were to start at a vigorous amount of weight which we can't handle. So how is this measured? As mentioned about the weight, so it's best to have a 1 to 2 warm-up sets first, just to start off nice and easy, get more blood flowing into your muscle quadriceps femoris, and then after that, progressing upwards. So it is recommended to have a 3 to 5 minutes of rest in between just to restore back our ATP PCR or adenosine triphosphate. We are using the RPE scale, which is the rate of perceived exertion, the box scale, 6 being the easiest effort and 20 being the hardest relative effort. So hopefully by the third or fourth set, then the individual would like to go off a RPE scale of 17 to 20. And how do we gauge that is to have at least a 50% of our own body weight. Yes, it is a highly reliable test for the 8RM across both genders, as it is a high test retest reliability and as well for the intensity for resistance training, according to Taylor and Fletcher in 2012. And finally, in summary, there is an increased muscular strength health outcomes if we were to do this exercise correctly and at the right frequency on average, as well as in terms of the muscular tone, flexibility, balance, and coordination. In addition to that, improved cognitive function and well-being. And as well, it is prevalent that it is a fantastic way to evaluate muscular strength tests and as well as reliability of the test. Thank you very much for listening to my presentation.
Thank you. I always like biomarkers I'm good at, and I'm good at leg extensions, so that's going to be one of my favorites. Tonight we have Myriam Grunewald, who's the research group leader and teacher at the Faculty of Medicine of Hebrew University in Jerusalem. She's also the founder and director of the Organoid Center at Hadassah Medical Organization, and we will be hearing about revisiting the vascular theory of aging. Thanks for joining us, Myriam.
Thank you, Brian, for the introduction and for the invitation to this webinar. I'm a big fan of this webinar, so it's an honor to be here. And I would like to thank also Joe Augustine from your team, who has designed this beautiful background that I think illustrates perfectly the fact that I am a vascular biologist. And I will show you today what we do as vascular biologists to understand the biology of aging and to try maybe to delay the onset of age-related disease from the lens of through the lens of the vasculature. And I will begin by, if I can, I will begin by quoting Thomas Seidenheim, who is a physician from the 17th century, who just by hearing the heartbeat of his patients and measuring their blood pulse, understood that a man is as old as his arteries. And by that, he initiated this kind of vascular theory of aging that says that the vascular health of an individual can be a very high driver in the pace of aging of the body of the individual. And maybe if we can maintain the vascular health, we could be able to impact the aging process of the whole of the different organs. And this is not very surprising for vascular biologists. It's quite obvious, but I think you don't have to be a vascular biologist to appreciate how this huge organ, and what you see here is a vascular cast of a young adult, and you can recognize the organs of this adult just by the vasculature that support these different organs. So the vasculature support the function of the organs, not only because they are tubes that distribute all the bloodborne substances, including hormones. They also clear the waste of our cells, and they are also the highway that is used by the immune system for immunosurveillance and entering the organs during inflammation. There is another reason that we think that the vasculature is also important for aging, is that the capillaries, which are these tiny blood vessels that are in contact with all the cells of our body, they produce secreted factors that have been coined endocrine factors and which are organ specific. And these endocrine factors support the organ during homeostasis and also participate actively in the regenerative process. For example, they maintain the adult stem cells in many of our tissues. And also they produce factors that really instruct parenchymal cells to re-enter the cell cycle to build up the organ back to its original mass. So what is aging if it is not losing this capability of recovering after an injury? So we think that the vascular aging is really poised to play an important, a critical role in the overall trajectory of the physiological aging. And we would like to show that if we maintain the vascular health, we could delay many or most of the age-associated diseases. So how do these vascular are aging? So I'm sure you are familiar with what happens with the macrovasculature, the large blood vessels. I'm sure you all heard about atherosclerosis that blocks the flow of the large arteries, or aneurysm, or other diseases that affect the large blood vessels and affect the flow and the perfusion to the downstream organs. But much less is known about what happens during to the microvasculature during aging. And the most prominent phenotype that we could measure is the decrease in what we call the microvascular density, the number of microvessels in an organ. And we could easily visualize by injecting fluorescent antibodies. And as you can see here, in 16-month-old mice, we already see a decrease, significant decrease, in the number of perfused microvasculature, for example, in the liver or in the muscle. And by the way, it is also true for human organs. Because, for example, in the skin of old people, we can visualize the perfusion, as you can see here, in a young aged person, aged 50, we also already see a decrease in the microvasculature density of the dermis. So this shouldn't happen because during evolution, we have evolved strong systems that when a cell senses a lack of oxygen or lack of nutrient, these systems are stabilized and they induced, among other things, the secretion by the stressed cells of pro-angiogenic factor that are secreted and instruct neighboring capillaries to grow and to restore a physiological perfusion. And one of these angiogenic factors is particularly interesting, the VEGF, the vascular endothelial growth factor, and he has been named the guardian of the vascular homeostasis by Professor Elie Keshet, my former mentor. And it is the guardian of the vascular homeostasis because first it is involved in all aspects of angiogenesis. from instructing the endothelial cells to disrupt the basal membrane, for migration of the endothelial cell, and so on. It is also a factor which is indispensable to maintain organ-specific endothelial cells phenotypes, for example, fenestration. And last but not least, it is indispensable to maintain an adequate capillary density in adult organs. And how do we know that? Because if we block VEGF signaling, for example, in cardiomyocytes of a young mouse, a 4-month-old mouse, after a while, which reflects the turnover of the capillaries, we see a decrease in microvascular density. And the perfusion will be restored only if we release VEGF blockade, which means that VEGF has no backup when in its role to maintain capillary density. So it seems that when we age, there is a problem with VEGF signaling. And indeed, when we checked it in different organs of aging mouse, we could see that the phosphorylation of the main receptor, the VEGF receptor 2, for angiogenesis decreased with age. And to cut a long story short, we didn't find any change about the VEGF, the ligand levels, but what we found is that we have many inhibitors of VEGF that are going up during aging, including a soluble receptor which acts as a decoy receptor for VEGF. It traps VEGF and doesn't allow its binding to the full-length receptor, and by that decreases the VEGF signaling. And by the way, this is also observed in the circulation of aged human beings. As you can see here, VEGF receptor 1 is going up with aging, both in men and women. So we had this idea that to neutralize this inhibitor, we could design a system that allows us to to increase a little bit the level of VEGF in the organs of these mice. Our system is a tetracycline-induced system controlled by tetracycline in the drinking water, and we can overexpress VEGF at will in any cell types in the body of these mice. And for this project, we use a liver-specific promoter. So in the same cage, it's a bitransgenic system. So in the same cage, we have both the mice that have elevated levels of VEGF and their counterparts that have just one transgene, so that they are not able to increase the level of VEGF. So in the same cage, you have both the treated animals and the control animals. And when we did this, we were able to modulate this system to obtain just a two-fold increase in the VEGF level in the circulation of these mice. And this is important because if you go too high in the VEGF in different organs in a mouse, you have deleterious effects such as permeability of the blood vessels, effect on the hematopoiesis, and so on. So we don't want this for the purpose of life extension and health path extension. The second thing that I want to draw your attention is that we begin this treatment at 8 months onward. So all the nice thing that I will describe to you is about maintenance of a young like phenotype and not reversing phenotype that are already established in an aged mouse. And when we did that, we saw that we were able to maintain a young-like level of phosphorylation of VEGF receptor 2, so VEGF signaling was maintained. And what is interesting is in the liver, but also in other organs, such as the muscle, remote organs. And just by doing that, we were able to maintain the microvascular density in all the organs that we have checked, including the liver, the muscle, the white adipose tissue, the brown adipose tissue, the bone marrow, and any other organ that we have checked. So we followed these mice with a bit increase in VEGF signaling. And we asked the question, what are the phenotypes or what are the diseases that this maintenance of microvascular density will impact? And the things that we have observed are, in my eyes, not less than amazing. Because to resume what we have found, the fact that we maintain the microvascular density has impacted in a positive way any of the system that we or organs that we have looked at. And because this has been published a while ago already, I will just resume the most important phenotype that we observed, and we'll move to some results that we have not yet published. So one of the most prominent effects by VEGF treatment was the observation that we have the maintenance of a young-like body composition and fat distribution. The body composition, we follow it by MRI on the same mouse for 10 or more months. And we also put them in echo MRI to measure the lean to fat ratio. And as you can see here, in the VEGF treated mice accumulate much less abdominal fat, which is important, but because this is a very inflammatory tissue. If we looked at the brown adipose tissue, which is maintained in old VEGF mice as compared to old mice, which gradually turn their brown adipose tissue into a white adipose tissue. And in the contrary, in the white adipose tissue in VEGF mice, we could see islands of brown adipose tissue. Interestingly also, the skin, the fat, the subdermal fat that disappears in old mice, also in old human beings, is maintained in VEGF-treated mice. So that's for the body composition and fat distribution. We put these mice in metabolic cage, and we could see that we maintain also a young-like metabolism. They continue to use mostly carbohydrates and not fat, and they maintain this metabolic flexibility. And by the way, these mice, they eat the same as the wild-type counterpart, but they don't gain as much as weight as the wild type do. So we entertain the idea that VEGF treatment could be a kind of exercise mimetic. The other thing that we did is to look at the liver of these mice, and we saw that the characteristic accumulation of lipid in the hepatocyte that are stained here in red in the liver of old mouse is really decreased in VEGF-treated mice, and these livers are less injured, are as measured by liver-specific enzyme in the circulation of these mice, such as ALT or AST. When we looked at the muscle, we saw that the mice are more performant on a Rotarot assay. They also have a better histology of their mouse, and their mitochondria are in a better shape and in a better function. When we looked at the bones of these mice, we were amazed to see the impact of VEGF treatment, both in male and female, that maintain a bone density equivalent to what we see in young adult mice. And maybe because of the good muscle and good bones, we could see also an improvement in the kyphosis of these mice, something that we also see in aging human. So all this and other phenotype that we have observed led to a very significant increase in the lifespan. But this, in my eyes, is not as important as the fact that the VEGF mice have a compressed morbidity period. This kind of a holy grail of the geoscience that is looking maybe to increase as a consequence of the compression of the morbidity period, to increase the lifespan. But more importantly, these mice were living much health. So we have succeeded in compressing this morbidity period. And I will take the last 5 or 10 minutes to draw your attention in one aspect of the phenotype that we have looked at, which is the tumor burden. Because when we speak about VEGF, people are saying, whoa, stop, because VEGF is highly correlated with cancer. And indeed, it is true. Cancer cells are producing tons of VEGF because they want to recruit more blood vessels in order to sustain their rapid growth. what we did is to follow all the mice that we used to construct this lifespan survival curve, and we carefully looked together with the histopathology department at the spontaneous tumors developing in our mice. And as you can see here, when we follow them with MRI, we could see that the mice at 2 years, this mouse strain developed a lot of spontaneous tumor, and we already saw in the VEGF gain of function mice that we have less of these spontaneous tumors. And when we measured that, we were amazed to see that not only we don't have more tumors, but we have less tumors that are spontaneously developing in these VEGF-treated mice, both in male and females. So we looked at what can do this wonderful thing that we develop less tumors. We saw that first we have less inflammation in these VEGF-treated mice. We have less age-associated immune infiltrates in the white adipose tissue, in the liver, or in the muscle, for example, that we could also measure by FACS. We also see that we have less that these inflammatory cytokines that reveal a low chronic level of inflammation in the aged mice, such as C-reactive protein or MCP1. Something very interesting is that we saw less of the senescence, for example, in the liver. And by the way, the cells that enter senescence in our mice, the first one are endothelial cells. We rarely see parenchymal cells that turn senescent. And when we measure them by beta-galactosidase assay and P16 measurement, we could see that if in an aged liver, we have a lot of endothelial cells that are senescent. In VEGF, we have much less of those cells, maybe because VEGF promotes an anti-senescent phenotype, or maybe we have a better system to take out these cells, these senescent cells, out of the tissue, of the organ. So we went after the immune system to see if we had this better cell that can take rid of these senescent cells. And Priya in the lab looked first at the thymus. And as you may know, the thymus in human and in mice is degenerating with age, as you can see here in this histology staining of the young thymus, and this is the functional part of the thymus, which disappears when we get old. And when we looked at the VEGF mice, we saw that the functional part of the thymus is maintained, even in very old VEGF-treated mice, which are very good news, because when we looked at the function, this is the total area, but when we looked at the function of these mice, in collaboration with the lab of Alon Montenegro in Ben Gurion University and Chesky Elia, we could see in the blood of the mice that we have more of these recent thymic emigrants and naive CD4 and CD8 cells that are circulating in the blood of these mice so that they can respond better against infection or other challenges. We also maintain a young-like peripheral immunity. Not only the thymus is maintained, but also the lymph node and the spleen show also an amelioration in these RTE or naive CD4 and CD8 cells. Something very interesting, aged mice and aged human accumulate these exhausted cytotoxic CD4 and CD8 cells. And as you can see here, we have a reduction, at least in the spleen, of the number of these exhausted T cells that are not able, because they express the PD-1, they are not able to target tumor cells and also senescent cells that were shown recently to express this PD-L1 ligand. So all this is also apparent in the T cell function that we looked at activation assay, proliferation assay. As you can see here, the T cells isolated from the spleen of a VEGF old mice do as much good as the T cells isolated from a young animal as compared to the old counterparts, and we are now in the process to challenge these mice with vaccines, SARS-CoV-2, to see if all these nice functions that we saw in vitro are also apparent in vivo in the mice. So for the sake of time, I will stop here, and I think that we can really revisit this vascular theory of aging. A man is old as old as his arteries, by a man and a woman are as old as their arteries and capillaries. And I would like to thank all the people that accompanied me in this very long study, our collaborators in Israel and abroad, and our funders. And thank you for your attention.
Thank you. It's a fascinating data. I was going to maybe start by asking some more simple questions because we don't generally talk about this tissue system that much in our webinars. You mentioned angiocrine factors. People have heard a lot on this webinar if they listen regularly about myokines and adipokines. But what's being secreted in the vessels? What are some of these factors and what are they doing?
Yeah, so as I mentioned in the beginning, we and others have shown that endothelial cells in capillary are important for two different things. One, they are the vascular niche of stem cells in adult tissue. We have shown that in the brain, in the region of the hippocampus, the dentate gyrus, where the brain stem cells, the residual brain stem cells are there. We have shown that in the hematopoietic system. Others have shown that in the testis, in many other tissue where you have stem cells. So they are important to maintain the stemness of the cells. And the other thing is that they secrete during homeostasis. They secrete factors. For example, the beautiful work of Shain Rafi, showing that in the liver, the endothelial cells are secreting factors that are important for the function of the hepatocytes. And when you injure the liver and the liver has to regenerate, they will switch to a regenerating mode. And these endothelial cells will secrete the factors that instruct the hepatocytes to re-enter the cell cycle, divide, until they reach the 100% volume of the liver. So these endothelial cells the capillaries are not only tubes that distribute the blood, but they are really instructing the parenchymal cells around them. And this is really organ specific with specific endocrine factors. And we are in the process of analyzing organ-specific endocrine repertoires. It takes a lot of time. It's variable because it really depends on the health of the mouse that we are testing. So we need a high number of that. But I put a lot of hope in this to decipher what happens in the endothelial cells when we age, beside all the things that we know about the endothelial cells in large blood vessels.
Yeah, no, you mentioned that there's a tissue-specific decrease in microvascular density, and you showed muscle and skin, maybe one other tissue. It looked liver, yeah, it looked very different, and so how much variation is there from tissue to tissue and changes in microvascular density?
So one of the things that
question. Yeah. Oh, go ahead and answer that first, and then I'll follow up.
Okay, yeah. One of the things that is really interesting is, organs are aging at different pace. Now we know that it's not all of the sudden that we are aging everywhere, every organ in our body, but we don't know in the mouse where the vasculature, the microvasculature begins to age. We have taken different time points to see how it evolves. So the liver, for example, vasculature is super resilient. I mean, until 14, 16 months, you have perfect liver vasculature. And then you can see a decrease in the microvascular density. In other organs, such as glands that produce protein hormones, you have fenestration. And these fenestrations are closing pretty early. in a mouse when we age. In the muscle, it takes also much more time to decrease, and it's different in female and male. So I think it is very important to study how these different microvascular beds will evolve in mice, and more importantly, in human beings. And we are now, through the organoid banks that we are receiving a lot of human tissue. And one of the things that we do is to analyze the microvascular density. And we hope that we will be able to make a more deep analysis of what is going on in different organs.
I think this is fascinating because, one of the probably underappreciated aspects of aging, I think, is the loss of stem cell regenerative potential in various tissues that occur during the aging process. And I wonder, you may not know completely the answer to this, but how much of a correlation there is between changes in microvascular density and loss of stem cell function with aging in various tissues.
Mm-hmm. Yeah, I know mostly study that we have done in the brain. We see that when we manipulate the vasculature and we increase the vasculature, then the vascular density in the dental gyrus, we see an increase in the stem cells that are also more functional, that they fire more. They can differentiate and they can fire more. In other system, in the hematopoietic system, the work of Sean Morrison and other people, and also we have done them. We have looked at if we manipulate the microvasculature, we can increase the number of stem cells without decreasing their repopulation capacity, which is nice. I mean, but I think that the key will be to analyze the endocrine factors in this vascular niche to understand really how they correlate with the loss of stemness.
You mentioned that there's changes in phosphorylation of VEGF receptor. And that's one of the big changes with aging. It made me wonder what the kinases are that are controlling that.
Yeah, so that's also a very good question. And we are trying now to do this for proteomics, but it's very hard to do that. And by now we don't have conclusive idea of what suddenly changed the phosphorylation of this. But what we think is that there are inhibitors that are sequestering VEGF, and then it doesn't allow the binding of VEGF to the full-length receptor and doesn't allow proper phosphorylation, because when we increase the VEGF a little bit, this phosphorylation is maintained. But you are right. Maybe there are downstream phosphoryl kinases that are impacting also this path. We don't know right now.
I wonder, just because there's so many of these signaling pathways that are linked to aging, that it may be able to tie up to some of the data on looking at those different kinases and aging pathways. Well, let's talk a little bit about the cancer issue, because that was you addressed it, but it was already on my mind before you addressed it. And you're right. There's this perception that cancer cells, well, it's not a perception. Cancer cells do enhance VEGF to try to recruit, angiogenesis, get a better blood supply. I mean, I guess the first question is probably a softball, but we think of cancer as an aging-related disease, and at the same time you have a decrease in microvascular density that's happening when tumors are most frequent. So does that mean that the density of the microvasculature may not be a driver in the initiation of the tumor, but then the recruitment of the microvasculature is important once the tumor is formed, or is that too naive?
Yeah, I think it's not too naive. I think the tumor really relies on the new blood vessels, and when you try to stop in the clinic VEGF signaling, the tumor will succeed generally in producing other angiogenic factors that will also recruit other blood vessels, because blood vessels are so important for the tumor growth. But for example, in pancreatic cancer, there is something interesting. The tumor is growing, but it's completely white when you take it out because the blood vessels that are there are not perfused at all. but they are important because these endothelial cells secrete endocrine factors that support also the tumor cells. So it's interesting. And the other face of this question is that what happens in metastasis? Because when you are aging, some cancer in aged people will be much more metastatic. And in most of the cancer, the metastases are going down, maybe because we have a decrease in these highways that the cancer cells are using. We have less blood vessels. But this is we would like to study this because it's not clear cut what VEGF will do, because on one way it increased the immune system, on the other way it increased the highway that the cancer cells can use. So it's a debate. But for spontaneous tumors in the mouse, we really see that there are less tumors in the develop spontaneously. Yeah.
I also wonder about, the distribution of VEGF. Like if you induce it in the liver, it's probably getting into the bloodstream and becoming pretty widely distributed throughout the body. Whereas in a tumor, you've got a localized induction,
Yeah.
I think, of VEGF in a lot of tumors,
Yeah.
which may be more restricted. And I wonder if that makes a difference as to whether, the VEGF is contributing to the tumor growth or not.
Yeah, I think you're right, but some tumors you can detect higher VEGF in the circulation of the patient for some really overexpressing VEGF tumors, and they impact the hematopoiesis. But I think you touch an important thing. What we propose is an increase twofold, not more than twofold of VEGF, not too much. And to induce or to increase the vasculature of a tumor, I think it will take much more than that. And by the way, we have in these mice that we have inoculated tumors also in young mice, not in old mice, and we couldn't detect a difference. When you have more VEGF in the circulation, it doesn't make you more make the tumor grow faster. So the mechanisms are different.
I wonder, that leads to a question. You mentioned that this is an induction in like a 10-month-old mouse, right? So yeah, young adult. I think that's what you said. What happens if you do it later? Like at 18 or 20 months where it be present.
Yeah, so we are trying that together with Merck company, Merck Israel. They ask us two things to do. Is one, to develop an alternative mode of VEGF delivery, not genetic, but another. We are doing AAV adenovirus associated encoding for VEGF, or liposomes with messenger RNA for decoding. We are in the process of doing that. And we begin at age 20 months. And what we have focused on the liver. We have designed an AAV that overexpress VEGF in the liver, and we focus on this very apparent phenotype of lipid accumulation. And I can already tell you that we have decrease in lipid accumulation in aged mice, which means that already when we have an aged fatty by expressing VEGF. And by the way, there is a paper that has been published recently doing the same thing, but in young mice put on. And here, and here also they have an established fatty liver, they induce VEGF, and they get a reversal of the phenotype. So these are very good news, because I can imagine that nobody will treat himself with VEGF for such a long time. I mean, if it would be wonderful to be able to reverse aging phenotype and not just avoid them. Yeah, both are nice, but if we can do shorter treatment when we are aged.
I mean, you may not know the answer to this, but so to see these effects, I wonder what the minimal period of induction is, to get to reverse, angiogenesis in the microvasculature and get all the signaling that you're talking about. What do you think that, could you do one treatment and for, with mRNA, or, it's an interesting question you may not be able to answer.
So with the virus, we can measure that, because we inject the virus and we can measure the VEGF in the circulation. and it stays for a long time because it's designed to stay a long time. And for 4 weeks, we don't have to re-inject. But interestingly, the effects are maintained. So I cannot present this data because these are very preliminary, but we know already that the turnover of the capillary is really long. For example, in the heart of a 4-month-old mouse, you have to wait for 6 months to see a decrease in microvascular density. Our blood vessels are resilient. So I guess that once, and we know also by our work that if you produce VEGF for 10 days, 14 days, it's enough to produce, to create stable blood vessels. If you induce VEGF and then you take it out before 14 days, the vessels are regressing because they are not covered by parasites and they are not mature. But for 2 weeks, it's enough to produce stable blood vessels. So our goal is to not to do constant injection with treatment with VEGF, but maybe once in a while, something like that, according to the turnover of the capillaries.
Just one last question. Are there sex-specific differences in the aging when you're in your mice where you induce VEGF?
Yeah. Blood vessels are really different between, and I was not aware about that until I really analyzed the transcriptome of the female endothelial cells and men. You have tons of estrogen receptors on endothelial cells, and they are very active in females. So we saw that in the lifespan, in the curve, which were with VEGF treatment, we saw really a rectangularization of the lifespan curve in the survival curve in female and less apparent in male. So I'm convinced that there are differences. We have to analyze it, and it's really on our to-do list. Yeah, in the brain, the vasculature is different in a male and female. It could explain a lot of things. Yeah.
I'm not going to comment on that. So, but thanks for doing this. It was wonderful having these questions. I'm going to bring in Weihan Hui, who's one of our really bright graduate students, and he's going to take some questions from the audience. Weihan?
Okay,
Okay,
thank you. Thank you, Brian. Thank you, Dr. Myriam, for your really inspiring talk. And we do have a lot of questions from the panel, so I'm going to pick the one that are not discussed in before. So the first question comes with 3 small questions. One is that, what are the primary genes that are upregulated or downregulated in a youthful vascular system compared to your older system besides VEGF? And also, are there other genes that are much less important than VEGF in aging? And also, have you ever looked at the partial epigenetic reprogramming of the vasculature to access a therapeutic potential to reverse aging?
Yeah. So these are very interesting questions. Unfortunately, I don't have any good response for that. The first question about what are the other, maybe other factors that are important. There was a paper that was published now that showed that basic FGF could be also important to maintain during aging and participating in the health of the vasculature. But I mean, there are other things. VEGF has been coined the guardian of the vasculature, but because really it is, without VEGF, you don't have good blood vessel. All our organs have a minimal expression of VEGF to maintain this vasculature. And regarding the epigenetic reprogramming, we would like very much to do that. And we were in contact with Campo. I don't remember his name, sorry, but who has this beautiful system that you can really induce the reprogramming in any cell types in the body. So we would like to try that in endothelial cells, and I guess it will give some interesting things. We have data on endothelial cells in collaboration with another lab here in Israel that makes epigenetic kind of clocks. And we see a difference and it's very interesting because the difference are in not in promoters, but in other regions of the genome that are very close to endothelial specific genes. So it's really nice to entertain the idea that there is changes in the epigenetic landscape in endothelial cells that may drive their loss of function, their gradual loss of function. But I cannot provide results on this, but I will. we are
Thank you
going
for
to
your
do that. There is a
Thank you
lot
for
to
your
do.
answer. Yeah. And the second question is that, so you mentioned that this VEGF might mimic this exercise. So do you have any evidence about the influence on VEGF by physical
Yeah.
activity?
So first of all, when you exercise, VEGF is going up. One of the problem in aging people when they exercise is that they can turn their muscle hypertrophic, but they cannot do sufficient vascularization. That's why it's very hard to increase the muscle mass in aged individuals. So for the muscle, what we saw is that for the mice, what we saw is that when we put them in metabolic cages, we saw that they eat the same, but they do not gain the same weight that the mice that are, in their cage. They don't do a lot of things, so they get big and fat. So the VEGF mice stay leaner. So they do not accumulate fat. They have a good mass of a lean mass of both bone and muscle. And also we looked at the mitochondria, and what we saw is that you have these two types of mitochondria in the muscle, which are in between the myofibril and the subsarcolemma, and we see a lot of very beautiful subsarcolemma mitochondria, which I saw is also accumulating in people that are training in a high level. So that's why it was funny to think that maybe we could do whatever we want, don't exercise, and eat whatever we want and stay thin and nice.
Thank you for your answer. So we do also see that there's a lot of question about the cancer issue. So another question is that could VEGF create a more favorable environment for cancer cells? And what about the pre-existing tumors? Have you ever tested about it?
Yeah. So we have done in young mice injection of tumors to model a pre-existing tumor, and we couldn't see a difference in this, but we didn't do that in aged mice. Maybe things will change. The only thing that we have looked at is the development of spontaneous tumors. And again, I think the levels of VEGF are different, and maybe the mechanism also is different. Yeah, the amount of VEGF that you have in a tumor doesn't provide good blood vessels. The blood vessels of a tumor are not healthy. They are permeable, and they are not. They are proliferating all the time. Our blood vessels are not. They are very stable. So I think the mechanism is different.
You mentioned
and the fact is that we didn't get more tumors in the mouse. Yeah.
Yeah, you mentioned that. Was there a change in the type of tumor?
What did you mean? I didn't understand.
Like there, you can get many different kinds of tumors with aging. Yeah. Was there a difference in the type of tumors that you observed?
So we have a list of the tumors that we have observed in our strain. Now we have moved to another strain, the BALB/C, and we could see the spontaneous tumor there are completely different. They do a lot of subdermal, huge mass of adipose cancer cells. So it's different. So it depends on the strain of the mouse and on the strain on the mouse. You have different. What we saw mostly is liver tumors in the strain that we have, lung tumors that are not very, but really, and a lot of leukemia. It's not unusual to see an aged mouse with a huge spleen. Leukemia, lymphomas, they are very common in this strain of mice, which are C57 Black 6, a substrain that we all have in Israel. Yeah.
Thank you. And the next question is that, so the growth hormones also maintains in the thymus, and it showed that the thymus is it doesn't shrink during aging with VEGF. So is growth hormone somehow related to this VEGF stuff?
Yeah, it's a good question. I don't know if we have a relation between growth hormone and VEGF. I will look at it. What we see is that with VEGF, the functional part of the thymus is maintained, yeah? And we have less of the adipose tissue that is invading the thymus in aging human and mice. So in the liver, we maintain more of the functional thymus, the immune cells, and the epithelium also that sustain the immune cells, the T cell.
Okay, thank you. So the next question is that, so since your controlled mice are looks relatively obese, so is there any evidence that the benefit of your VEGF go beyond the benefit that would be expected from the weight loss induced by the VEGF?
Nice question. The question that we always ask is, what is the organ that VEGF is ameliorating that may be the driver of this extended lifespan and extended lifespan? Because all the organs are connected. I don't know if I understood your question, but maybe it's the weight loss, maybe it's the liver that is better, maybe the muscles. With all these myokines and adipokines and all these connection between all the organs, it's difficult to pinpoint which phenotype drives the other phenotype. But this is also an interesting aspect of geoscience to understand if we have something like a hierarchy in the aging organs.
I agree. I think that's one of the most fundamental questions in geroscience, and it can be any intervention that extends lifespan. I don't think we've really answered that yet, even for things like mTOR inhibitions.
Yeah.
Thank you. And the next question would be like, how does all this related to the age-related macular degeneration?
The macular degeneration, yeah. So in macular degeneration, you have the contrary. You have the VEGF levels. You are talking about the wet macular degeneration, right?
Mm-hmm.
Yeah.
I think
so
that
you have this variation of VEGF levels in the eye, and the blood vessels are growing, and then VEGF levels are going down, and then the vessels are regressing. So there is a mess in macular degeneration, and indeed the cure is to inject a VEGF inhibitor. Yeah, but the thing is that ophthalmologists are now following the people that have been treated with Avastin, which is a VEGF inhibitor, for 20, 30 years. And there is an effect on the vasculature of the eyes of these people. And there are claims that it also impacts other vascular beds in the vicinity of the retina. So, yeah. So this is a typical disease when overexpression of VEGF causes the disease. But if you treat with anti-VEGF for a long time, you also have a decrease in the vascular density, because VEGF is important to maintain the vascular density.
That was one of my questions. If you treat, that's a localized treatment, I think, but if you treat systemically with a VEGF inhibitor, are there side effects that you might see that are associated with aging, or is the treatment just so short that you don't see that?
Yeah, so we have the reverse system where we can overexpress a soluble form of the VEGF receptor, which acts as a decoy receptor. So it's a kind of expressing an inhibitor of VEGF. And it is very interesting because when we built this model, we thought that, okay, we will induce it in 4 months old and we will have aged mice. Okay, because we decrease the microvasculature, and we have to wait a very long time to see an effect, to see a phenotype. We begin the VEGF signaling blockade at 4 months, and we wait until 14 months to see an effect in the liver, an effect in the immune system, which is beautiful. I mean, we also have done LPS challenge in these mice where VEGF is blocked, and we see that they die instead of coping with LPS like the other old mice. And so when we block VEGF, we don't have a premature aging phenotype, because really the endothelial cells are resilient. These are cells that are extremely resilient because they are so important, and we have to wait a long time to have an effect. Maybe cancer people that receive a high dose of Avastin, they may have other side effects of this, but they are already ill. So it's very difficult to understand what is the tumor impact on their aging phenotypes or the soluble fit itself.
Thank you. One last question. So do you observe an induction of nitric oxide production by these endothelial cells following the...
Yeah, VEGF is induces NO. Yeah.
I see. And also, do you think the level of nitric acid production by the endothelial cells could delay the vascular aging?
Yeah. Could also be something worth to check. Yeah. We are recruiting, by the way.
Well,
If some student want to come to Israel to join us, we have a lot of things to do. So I mean, it's not maybe the best period to come to Israel, but I guess that in a couple of months, all this situation will be resolved. Yeah. We have a lot of, and you ask very
hope so.
interesting. I mean, yeah.
Well, thanks a lot for joining us, Miriam, and thanks, Weihan, for doing all the questions. We'll have you back get an update at some point. I want to remind participants to use the chat function, and panelists and all attendees option to leave comments and feedback on the show. Next week, we'll have Kenneth Meyer, who's a professor and group leader of Human Movement Sciences Research Unit at Maastricht University. We don't have his title, but I think by virtue of his position, we can guess a little bit about what he's going to be talking about. And then we have a lot of announcements coming up for the Center for Healthy Longevity and programs in the U.S., including a Healthy Longevity Intensive course in 2025 for healthcare professionals, the Founders Longevity Forum Singapore Unlock Healthy Longevity event, which is February 27th and 28th. And that's designed to bring investors, innovators, entrepreneurs, and researchers in biotech and consumer longevity. You can register for that. There'll be another 2025 Healthy Longevity Talent Incubator coming up, 10-day intensive course in healthy longevity. And also, we haven't officially announced this yet, maybe next week, but we're going to have a conference on the physics of aging in Singapore in March. And so that's really about how do we understand aging from a more dynamic system point of view, and how do theoretical modeling, which physicists are really good at, how can we apply that to aging? Before I go, I want to again thank you for joining us and leave you with a final video about asking our seniors how they stay active and healthy.
We're here at Eunos Crescent to interview some elderly for some health advice. What do you do to keep healthy?
I do exercises and try to take less salt meals.
Exercise. Every morning, go for a walk before the sunrise.
Yoga, Zumba, Sudoku. Sudoku.
That one is for the mind.
Sudoku.
And bingo is the most.
And bingo also.
出去聊聊天聊聊天呐，开心点啊，不要想这样多。
I try to eat healthy lah. Sometimes orange, sometimes watermelon, sometimes papaya, mango.
How about vegetables?
Vegetable I cannot because I no teeth. I cannot bite.
I'm keep healthy and keep fit. I have to do exercise. I'm doing exercise 3 times a week.
I used to go down to a fitness park to do my own fitness. I do all calisthenic exercises. I only rest only Sunday.
We are aging, so we need to make ourselves active so that we can have a longer life, right?
I'm feeling like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down.